Okay, let's unpack this. Welcome to the deep dive.
And today we are going right to the architectural heart of, well, of all vertebrate life.
We really are.
We're diving deep into the mesoderm, that foundational, absolutely crucial middle layer that determines the entire structural and functional body plan.
Exactly. I mean, just imagine for a second one single embryonic layer that's responsible for basically everything structural and functional in your body.
Everything.
The bones holding you up, the muscles that let you move, your heart pumping your blood, the kidneys cleaning it, it's all mesoderm.
It is.
So our mission today is to trace the molecular blueprint of this layer. We want to reveal these precise, repeatable signaling rules, the molecular clockwork, the chemical gradients, and the essential communication loops that transform a simple sheet of cells into these incredibly complex, functional organ systems.
And this deep dive, it's really all about cause and effect. We're going to see how tiny molecular oscillators can determine the precise number of segments in your spine.
Which is just amazing.
It is. And how, a specific concentration of a single signaling molecule can literally pattern your fingers. And how two tissues have to engage in this kind of conversation, a reciprocal induction, to successfully build a functioning kidney.
It's a remarkable study in developmental precision. So before we jump into all the details, maybe we should quickly review the geography of the mesoderm, because where a cell is located, that really dictates its destiny.
Absolutely. To set the stage, you have to recall that the mesoderm partitions pretty early on into four major zones, and this is moving from the center of the embryo outwards.
Medial to lateral.
Exactly. We start right at the midline with the notochord. Now, it doesn't form structural elements itself, but it acts as this critical signaling center, basically telling everyone else what to do.
It's the conductor of the orchestra.
That's a great way to put it.
Yeah.
And flanking the notochord is the paraxial mesoderm. This is our segmentation engine. This is the tissue that will divide into those paired blocks called somites.
Okay, then moving further out, laterally, we find the intermediate mesoderm.
Right, which has the very specialized task of forming the urogenital system. So that's the gonads, the kidneys, and the adrenal cortex.
And finally, the most lateral region.
That's the lateral plate mesoderm. And this is a big one. It splits into two layers. And from it, you get the entire ventral body wall, the limb buds, and the entire circulatory system, blood vessels, the heart. The sheer functional diversity coming from this one layer is just astounding.
It really is. And we're going to begin our deep dive today by looking at the most fundamental structural feature of the vertebrate body axis. The segments, which, as you just said, are built by the paraxial mesoderm.
Let's do it.
The precision of this early segmentation is, well, it's almost unbelievable when you first learn about it. We're talking about somitogenesis, which is the process where these blocks of tissue, the somites, form sequentially along the embryonic axis.
And they always form from anterior, so the head end, to posterior, the tail end.
And the rhythm is genetic. It's not random at all. The number of these somite blocks is a totally rigid, species-specific trait.
It is. A chick embryo, for instance, will form a new somite with clockwork regularity every 90 minutes.
90 minutes on the dot.
A mouse takes a little longer, maybe 120 minutes. But then some fish species, they can just crank them out every 30 minutes. It's like a rhythmic, segmented, automated production line.
So what's the first physical step? What can we actually see happening?
The initial step involves the cells condensing. They go from being this loose association within the segmental plate of the paraxial mesoderm, and they pack together into these tightly formed epithelial vesicles. It's a literal change in their cellular infrastructure.
So they go from loose to tight. What's driving that change?
That change, that mesenchymal to epithelial transition, is driven by an increase in cell adhesion molecules. Specifically, the cells dramatically upregulate the expression of fibronectin and this crucial adhesion protein called N-cadherin.
N-cadhere.
Yeah, you could think of it like cellular Velcro. It binds the cells tightly together to form that distinct, observable epithelial border. And that tight epithelial somite is really the first visible sign of segmentation.
Okay, but the big question is, what dictates this perfect rhythm? And the exact location of the segment boundary. That's where the sock and wavefront model comes in.
It does. It's such an elegant model.
It really is. It's a verbal diagram that beautifully illustrates how time and space interact to define where one somite ends and the next one begins.
And you need two main components for it to work, operating in perfect harmony. You need a clock, which controls the time, and you need a gradient, which controls the space.
So let's start with the clock, the temporal element.
Right. This is an intrinsic molecular oscillator, and it's built primarily using components of the Notch pathway.
And the key players here, the key molecules, are the Hes genes. These encode what are called basic helix loop helix transcription factors, or BHLH for short. And you also have accessory proteins like lunatic fringe.
And these Hes proteins are the ultimate molecular metronome. The reason is they are inherently unstable and they repress their own transcription.
Ah, so it's a negative feedback loop.
A perfect molecular negative feedback loop. So, Hes protein levels rise inside the cell. When they hit a certain concentration, they move into the nucleus and they turn off the very gene that made them.
But because the protein is unstable.
It rapidly decays, the repression lifts, and the gene turns back on, starting the whole cycle over again. And this cycle of repression and decay creates this rhythmic oscillation of mRNA levels that corresponds exactly to that 90-minute or 120-minute period required to form one new somite.
That is just an astonishing feat of molecular timing. But timing alone isn't enough, right? If every cell in the tail bud was just oscillating, how would they all know where to, cut off the next segment?
Precisely. You need spatial control.
And that's where the gradient, the wavefront, comes in.
Exactly. The spatial cue is a concentration gradient of fibroblast growth factors, or FGFs, mainly FGF4 and FGF8. This gradient extends posteriorly along the axis. The highest concentration is at the very tip of the growing embryo, and it gradually decays as you move up toward the head.
Okay, so the beautiful synthesis here is the integration of these two signals, the clock and the gradient.
Yes, the wavefront is simply the location where that FGF concentration falls below a crucial permissive threshold.
So when the cells pass this threshold and they experience this low FGF level, the molecular clock, which is still rhythmically oscillating, can finally act.
It can. It acts to upregulate the transcription factor, MES2.
And MESP is the trigger.
MESP is the molecular trigger for segmentation. When that Hes clock reaches its permissive phase and the FGF level is low enough, MES2 stabilizes a steep fringe gradient, which in turn activates Notch signaling. And that signaling cascade then drives the crucial cell adhesion changes required to physically segment the tissue.
So it turns off the genes that keep cells loose and turns on the cadherins.
Exactly. It represses genes like snail, which normally prevents cell adhesion, and that permits the upregulation of cadherins and the formation of a tight epithelial segment boundary.
So if you visualize this for a moment, the clock is always running in the background, but the starting pistol for actually forming a segment can only fire when the cell passes that wavefront, when the FGF concentration drops below a certain level.
It's the perfect harmony between a temporal rhythm and a spatial boundary.
And we see the devastating clinical relevance when this clockwork is just slightly off.
Oh, absolutely. Many forms of congenital scoliosis, curvature of the spine, are directly linked to mutations in these segmentation genes, things like delta-like 3, lunatic fringe, and particularly MESP2. A slight timing error in this molecular system creates segmental abnormalities that lead to severe spine deformation.
Which makes perfect sense. Once a somite is formed, though, it has a very short window before it starts specializing. And what's interesting is that its eventual fate, what it becomes, is not intrinsic. The decision isn't made within the somite itself.
Right, we know this from these classic, really elegant microsurgical rotation experiments done in chick embryos.
What did they do?
Well, they took newly formed epithelial somites, and they either rotated them 180 degrees or moved them to a totally new location. And they still differentiated correctly relative to their new surrounding tissues.
So that proves they're listening to their neighbors.
It proves they rely heavily on inductive signals from the notochord, the neural tube, and the epidermis.
Okay, so the first major differentiation event is the formation of the sclerotome. This happens on the ventral and medial side.
And this tissue is induced primarily by that key signal that defines the ventral axis of the embryo, sonic hedgehog, or shush.
Which is released from the adjacent notochord and the ventral neural tube.
Right. So when these sclerotome cells receive they immediately undergo an epithelial to mesenchymal transition, or EMT. They break those end-cadherin junctions we just talked about and revert back to being loose mesenchymal cells.
And as they transition, they upregulate a new set of transcription factors.
They do. Things like PAX1, PAX9, and NK by 3.2. And crucially, they also express high levels of ID-class myogenic inhibitors.
Myogenic inhibitors, so they block muscle formation.
Precisely. These inhibitory proteins are essential because they block the very factors that would turn a cell into muscle, ensuring that the sclerotome's fate is purely skeletal.
So where does the sclerotome end up? What does it become?
The ultimate fate of the sclerotome is to form the cartilage skeleton of the trunk. So we're talking about the vertebrae, the ribs, and the dorsal part even forms the tendons, which is sometimes called the syndidome.
Now, what's genuinely fascinating and a point that I think often gets missed is the mechanics of how the vertebrae form. It involves this necessary realignment called resegmentation.
That's right.
You might just assume one somite forms one vertebra, but that's not the case at all.
It's not.
Yeah.
Each finished vertebra is not derived from a single somite. Instead, the posterior half of one sclerotome fuses with the anterior half of the next, more posterior sclerotome. This actually shifts the segmental boundary.
Why the shift? I mean, why is that necessary?
It's fundamental for motor control. It ensures that the spinal nerves, which have to grow out into the body wall, end up aligned perfectly between the resulting vertebrae.
Ah, of course. If the spine didn't e-segment, the nerves would be blocked by solid bone. You'd have no motor function.
Exactly. It's a very elegant solution to a mechanical problem.
And the identity of those vertebrae, whether they become cervical in the neck, thoracic in the rib cage, or lumbar in the lower back, that's not arbitrary either. It's specified by the regional combinations of Hox genes that are expressed at that level.
Yes, think of Hox genes as the axial zip code.
It's a beautifully conserved code, too, even if the absolute number of vertebrae changes between different species.
That's right. For instance, the anterior boundary of Hox6 expression consistently marks the transition point between cervical and thoracic vertebrae, whether you're looking at a mouse or a human. It's the same landmark.
Okay, so moving to the other major fate, the remaining lateral part of that epithelial somite forms the dermomyotome.
Right, which then splits again into the dermatome, which will form the dorsal dermis that's induced by neurotrophin 3, and the myotome, which is the precursor for all skeletal muscle, the process we call myogenesis.
And myogenesis is complex because muscle arises from different regions via slightly different molecular routes, which then converge later on.
Exactly. We have the epaxial myotome, which is medial, and forms the deep segmented muscles of the back right along the axis. And then we have the hypaxial myotome, which is lateral, and forms the ventral body wall, the limb, and the diaphragm muscles.
Let's start with the epaxial pathway, the one closer to the midline.
Right. It requires a two-step induction sequence. First, it needs that early exposure to shh that we talked about, followed by a strong want signal coming from the dorsal neural tube. This combined signal is what turns on the key myogenic gene MyF5.
So it's not just one signal, it's like a two-factor authentication system. But there's a vital balancing act here, right, involving another player.
A very important one, BMP4, which is diffusing in from the adjacent lateral plate mesoderm. BMP4 normally acts as a powerful repressor of myogenesis. If you don't block it, you simply don't make muscle.
So how does the system block it?
Well, the system cleverly blocks it. The Wnt signal, in addition to activating MYF5, also induces the expression of noggin, which is a well-known BMP inhibitor right in that medial region.
So noggin forms a shield.
It effectively blocks the repressive BMP4, setting a precise boundary that permits the epaxial myotome to form muscle, while areas outside that noggin zone remain suppressed. This antagonism is how the boundary of the muscle field is defined.
Meanwhile, the hypaxial myotome, which is further away and more lateral, needs a slightly different set of signals.
It does. It needs 1T7A from the dorsal epidermis, plus a permissive but not inhibitory level of BMP. And these cells are different because they express specific transcription factors like LBX1 and the receptor c-Met, which, as we'll see later, are absolutely crucial for their great migration into the limbs.
Okay, so ultimately, all these different signaling pathways, they converge on the same family of powerful myogenic master switches.
They do. These are the BHLH transcription factors, MyoD, Myf5, myogenin, and MRF4, which is also called MYO6.
And they are called master switches for a very good reason.
Yes. There were experiments that showed if you took an ordinary non-muscle cell, like a fibroblast, and you forced it to express one of these genes, it would commit and turn into a muscle cell. It proves they are sufficient to just flip the cell's fate.
So how do they work, mechanistically?
Their mechanism relies on forming heterodimers with common nuclear proteins called E proteins. But remember the sclerotome. It expressed those IDE-class myogenic inhibitors.
Right, the things that blocked muscle formation.
Well, these inhibitory HLH proteins work by forming unproductive inhibitory dimers with the MyoD factors. They essentially sequester them and ensure that non-muscle cells like bone cells stay non-muscle cells.
That's why the sclerotome doesn't suddenly sprout muscle fibers. It's actively repressing the signal. And what about redundancy? You mentioned MYF5 and MyoD.
Right. They show a surprising amount of functional redundancy. If you knock out just one of them, the animal is mostly fine. But the fact that the double knockout eliminates skeletal muscle entirely proves their essential combined role in initiating the entire lineage.
And there's a hierarchy here too, right? With myogenin.
Yes, myogenin acts downstream of MyoD and Myf5. It's essential for the later stages of muscle formation, specifically for differentiation and fusion. So, a knockout of myogenin leads to severe muscle deficiencies, even though the initial commitment signal was there.
Okay, so once a cell is committed, it's called a myoblast. These myoblasts have to stop dividing, they become postmitotic, and then they engage in one of the most visible steps. They fuse together.
They do. They form these large multinucleated structures we call myofibers or myotubes. And this isn't just simple merging, it's a very choreographed event.
What's involved in that fusion process?
It involves complex molecular recognition at the cell surface. It utilizes cell surface molecules like the Kir-like homologs you see in zebrafish, and also the activation of specialized enzymes like metalloproteases, specifically the ATOM class, sometimes known as miltrins.
And there's also a requirement for calcium signaling.
Absolutely. A rise in intracellular calcium activates the transcription factor NFETC2, which is necessary for proper myoblast fusion. Mice that lack NFETC2 still form muscle, but they have smaller, weaker fibers with fewer nuclei, confirming its critical role in the fusion mechanics.
And this whole process, thankfully, doesn't stop at birth or after an injury.
No, muscles rely on specialized, persistent progenitor cells called muscle satellite cells. These are mononuclear cells. They have a single nucleus, and they reside quietly under the basement membrane of the mature myofibers.
And they're characterized by the expression of the transcription factor PAX7.
Right. These PAX7 positive cells are the primary agents responsible for all postnatal muscle growth and, critically, for adult repair following damage. They give muscles their regenerative capacity. They are the sleeping builders, ready to activate when the muscle is strained or damaged.
That really brings the story of the segmented mesodermal axis full circle, from the rhythmic creation of the somites to the final specialized creation of skeletal muscle and bone.
It does. Shall we move lateral now into the intermediate mesoderm to look at an organ that really epitomizes this idea of precise reciprocal induction? The kidney.
Let's do it. The kidney is, well, it's arguably the most essential filtering mechanism in the vertebrate body. Developmentally, it's a classic model because it cannot form by simple self-assembly.
No, it requires an explicit molecular reciprocal inductive interaction between two entirely distinct tissues to form its mature filtering structure.
So if we look across amniotes, that's reptiles, birds, and mammals, kidney development proceeds through 3 evolutionary stages in sequence.
That's right. You have the vestigial pronephros, then the transient mesonephros, and finally the definitive adult kidney, the metanephros.
And we're going to focus on the metanephros, which houses the functional unit, the nephron.
And a nephron is this complex microscopic structure. It involves the renal corpuscle, that's the glomerulus inside Bowman's capsule, where blood is initially filtered, which is then connected to this vast network of convoluted tubules and eventually the collecting ducts.
So to build this, the definitive kidney needs 2 main components, and both are derived from the intermediate mesoderm.
Correct. First, you have the epithelial component, which is the nephric duct, specifically an outgrowth from it called the ureteric bud. This is going to form the entire collecting duct system.
And the second component
Is the surrounding loose tissue, the nephrogenic mesenchyme, which will form the actual filtering nephrons themselves.
And it truly is a story of cooperation. I mean, if you separate the ureteric bud and the mesenchyme in a petri dish, they just sit there and do nothing.
They must interact physically and chemically. And before that conversation can even start, the mesenchyme has to be ready. This tissue must express the zinc-finger transcription factor, WT1, that's Wilms tumor 1, to be considered competent to respond to the bud signals.
And the critical nature of this readiness is shown so dramatically in clinical terms.
It is. A knockout of the WT1 gene, or mutations leading to its loss of function, results in a complete failure of metanephric kidney development. This factor is the molecular key that unlocks the mesenchyme's potential.
Okay, so once it's competent, the first signal of this reciprocal induction occurs. Induction 1, mesenchyme to bud. The mesenchyme dictates where and how the epithelial collecting system is going to grow.
And the key signaling molecule here is GDNF, glial-derived neurotrophic factor, which the mesenchyme secretes. GDNF is essentially a homing beacon.
And the ureteric bud receives this beacon through its specific receptor, a tyrosine kinase known as RET.
And this GDNF pathway is arguably the most essential molecular pairing in kidney formation. It provides the chemotactic signal that drives the ureteric bud to grow into the mesenchyme and, crucially, to branch extensively.
And when we say extensively, we mean structurally complex. In a mouse kidney, this branching happens for about 10
10
generations.
generations deep, yeah. It creates the vast network that forms the renal pelvis and the entire collecting duct tree. The necessity is absolute. Knockouts of either the GDNF gene or the rate receptor abolish kidney formation entirely.
And conversely.
Conversely, RET gain-of-function mutants show this unregulated chaotic ureteric bud growth because all the normal controls are lost.
So once the bud has grown in and is branching, it immediately signals back to the surrounding tissue in induction 2, bud to mesenchyme.
Right, the bud essentially tells the mesenchyme, okay, I'm here, start building the filters.
And the inducing signal from the epithelial ureteric bud is believed to be WNT9b.
Exactly. And this WNT9b signal delivers the command that causes the nephrogenic mesenchyme to undergo its dramatic reversal, the mesenchymal to epithelial transition, the ME transition.
This ME transition is really the cellular cornerstone of nephron formation.
It is. The loose mesenchyme first aggregates into these dense, tightly packed caps around the tips of the bud. These caps then form epithelial vesicles, which then morph into recognizable comma-shaped and S-shaped bodies, and these eventually elongate into the complex, convoluted tubules we associate with a nephron.
And this is more than just a shape change. It's a total overhaul of the cell's identity at the molecular level.
It is. For instance, the cells switch their extracellular matrix components. They stop making mesenchymal things like collagen I and third and start making epithelial components like laminin and collagen IV to lay down a proper basement membrane.
And they also swap out their cellular adhesion structures.
Right, replacing things like NTI with a strong epithelial adhesion molecule, ecadherin, which locks them into that tube structure. And the WNT9b signal is upstream of a crucial upregulation of Wnt4 in the aggregating mesenchyme, which is also required for the subsequent elongation and differentiation of the tubules.
And this whole process of aggregation and transition requires constant life support.
It does. FGF2 and BMP7 are released by the epithelial bud specifically to ensure the mesenchyme cells survive and aggregate during tubule formation. Without them, the mesenchymal cells just undergo programmed cell death.
We can see the necessity here again, BME7 knockouts, for instance.
They result in dysplastic, poorly formed kidneys where the nephrons are blocked at that early comma or S-shaped stage, and they fail to fully elongate.
Finally, we have to consider the population that sustains this growth.
Yes, the cap mesenchyme expresses the transcription factor 6-2, and these 6-2 positive cells are the nephron progenitors. They maintain the capacity to generate new nephrons through division and differentiation.
This brings up a really profound question about the loss of regenerative capacity in mammals.
It does. In mice, these 6-2 positive progenitor cells persist and divide until just shortly after birth, making new nephrons. But after that critical window, they are lost, and the capacity to make new nephrons is gone forever.
Which is in stark contrast to lower vertebrates.
Like the zebrafish, yeah. They retain this progenitor pool throughout their entire life. They can continue to make new nephrons and regenerate damaged kidney tissue. It's a crucial difference between our developmental strategies.
That mechanism, mesenchyme signaling epithelium, which in turn signals back to the mesenchyme, is the blueprint for so many organs, but it's just so clear here in the kidney. Let's stay within the intermediate mesoderm now, but let's look at the process of establishing the germ cells and the gonads, the ultimate sex switch.
Gonadal development involves this essential biological duality. On one hand, you have the somatic structural tissues of the gonad, the cells that will form the testes or ovaries, and these come from the genital ridges, which are derived from the intermediate mesoderm.
But the gamete-producing cells, the primordial germ cells or PGCs, they originate far away in the embryo's posterior and have to migrate to their final destination. Their journey is one of the earliest examples of cell lineage commitment.
Right. In the mouse embryo, PGCs are induced in the proximal part of the egg cylinder by strong signaling molecules, BMP2, NF4, and NF8b, which are released from the adjacent extraembryonic ectoderm.
And once they get that signal, they become fully committed.
They do. And this commitment is marked by a specific molecular fingerprint. They repress somatic genes using the zinc finger factor BLIMP1, while at the same time persistently expressing core pluripotency genes like OCTI4, NANOG, and SOX2.
So it's a dual action locking down the non-germ cell fate while maintaining their potential to become gametes.
Exactly. And once they're specified, they have to embark on this pretty treacherous journey. They travel from the base of the allantois, along the hindgut mesentery, all the way to their definitive location, the genital ridges.
And this migration is strictly controlled by a chemical breadcrumb trail, a process known as chemotaxis.
It is. The guiding signal is the chemokine STF1 stromal cell-derived factor 1, which is expressed by the lateral plate mesoderm, and this is detected by the receptor CXCR4, which is expressed on the PGCs themselves.
And this guidance is non-negotiable.
Completely. Loss of the CXCR4 receptor means the PGCs can't read the breadcrumbs, and they fail entirely to colonize the genital ridges, which results in sterility.
So while they're migrating, the genital ridges themselves, the somatic framework, are developing autonomously.
Yes, and they require the early presence of some key transcription factors. WT1, the same one that was critical for the kidney, and SF1, which stands for steroidogenic factor 1.
And as the name implies, SF1 is there to lay the groundwork for hormone production later on, even when the gonad is still indifferent.
Right. This early period, before the sex is determined, is called the indifferent gonad stage, and it's characterized by the presence of two parallel duct systems in both sexes, just waiting for the switch.
So we have the Wolffian duct, which structurally is the leftover nephric duct, and that will persist and form the male internal genitalia, the epididymis, and vas deferens.
And right alongside it is the Mullerian duct, or paramesonephric duct, which will persist and form the female internal genitalia, the oviducts, uterus, and vagina. Both are present until about embryonic day 12.5 in the mouse.
So then comes the critical moment of divergence in mammals, the biological sex switch, and it's the expression of a single gene, SRY.
The sex-determining region of the Y chromosome, and the evidence that SRY is the master switch is just overwhelming and clear. Genetically male mice, XY, that lack SURE develop as females.
And conversely, XX mice engineered to express SURE
develop as males. It is a single, elegant but crucially transient gene expression that flips the entire developmental trajectory.
Okay, let's follow the male pathway. SRY-onin. SRY is expressed for only a short period.
But that burst immediately activates the persistent expression of SOX9. And SOX9 is the high-level regulator that locks in male development.
And SOX9 drives the differentiation of two key cell types.
Yes. First, the Sertoli cells, which organize the gonad into seminiferous tubules and support the germ cells. And second, the adjacent mesenchymal cells differentiate into Leydig cells, which are the producers of the male sex hormone, testosterone.
But the male body also has to actively destroy the female system.
It does. SOX9 starts that process by upregulating AMH, which is anti-Mullerian hormone, a member of the TGF-beta superfamily. And AMH acts specifically to cause the Mullerian duct to degenerate.
And the continued presence of SF1 sustains both AMH and testosterone synthesis, which in turn drives the formation of male secondary characteristics and ensures the Wolffian duct persists.
Right, developing into the vas deferens and epididymis.
Now, in the female pathway, SRY-OFF, the system just defaults to ovarian development.
It does. The absence of SRY allows the persistent expression of key ovarian factors, namely Wnt4 and FOXL, in the differentiating follicle cells. And these factors actively repress any stray SOX9 activity.
And Wnt4 persistence is actually required for subsequent ovary development.
It is. And since AMH is never produced, the Mullerian duct is not suppressed, so it persists to form the oviducts, uterus, and vagina, while the Wolffian duct just degenerates from a lack of hormonal support.
And here is that fascinating twist you mentioned earlier. The fate of the germ cell itself is determined entirely by its surrounding environment.
It is. Any PGC that fails to enter the gonad, it doesn't matter if it's genetically XX or XY, it will develop along the female pathway as a new site.
That's massive. So a cell's entire sexual destiny is determined not by its own X or Y chromosomes, but by the molecular environment of the tissue it lands in.
It just proves how dominant the somatic organizing tissue is in establishing the developmental pathway.
It's a perfect setup for our next section. Just as the somatic cells organize the germ cells, we are now going to pivot to another major structure from the mesoderm, the limbs, which requires coordination between three separate signaling centers to pattern a three-dimensional appendage.
The development of vertebrate limbs, whether they become arms, legs, wings, or fins, is one of the most thoroughly studied systems in developmental biology. It serves as a near-perfect model for understanding how morphogen gradients pattern complex three-dimensional structures.
And all limbs start as these simple bulges on the flank called limb buds.
Right. And a limb bud has two main structural components: a core of mesenchyme, which is derived from the somatic lateral plate mesoderm, and will form the skeleton connective tissue, and that's covered by a thin epidermal layer.
So to initiate the limb bud, a signal has to first commit the somatic mesoderm in a specific location.
And that initial signal is provided by Wnt signaling. It's WNT2B for the forelimb, WNT8B for the hind limb. And this upregulates the crucial growth factor gene FGF10 in the mesoderm right beneath the prospective limb area.
And FGF10 is the starting gun.
It is. It satisfies the criteria of necessity. If you look at an FGF10 knockout mouse, it forms no limb buds at all. The expression of FGF10 is the molecular prerequisite for limb outgrowth.
And the identity of that limb forelimb versus hind limb is specified by different members of the T-box gene family.
Exactly. TBY5 marks the forelimb and TBK4 marks the hind limb. And both of those are regulated by the local Hox gene code at that axial level.
And while the skeleton and connective tissue come from that somatic mesoderm core, we can't forget the muscles.
Right. The myoblasts don't originate in the limb. They have to migrate in from the adjacent somites we talked about earlier.
And this migration is guided by a chemical signal from the limb mesenchyme called HGF, or hepatocyte growth factor, also known as scatter factor. It acts chemotactically.
And the myoblasts, remember those hypaxial cells? They express the MET receptor, which binds HGF, and that directs them precisely to the growing limb.
So now for the big challenge. How does the embryo manage to pattern 3 dimensions at once? The complexity of the limb is built by patterning it simultaneously along 3 distinct perpendicular axes.
Proximal distal, so from shoulder to fingertip, antero-posterior, from thumb to pinky, and dorsiventral, from knuckle to palm.
Let's start with the lengthening axis, axis 1, proximal distal PD outgrowth. This is driven by the apical ectodermal ridge, or the AER.
Right, a specialized, thickened strip of epidermis found right at the very tip of the growing bud, initially induced by the underlying mesoderm. The AER acts as the master regulator of outgrowth. It's basically the gas pedal.
And it secretes a continuous potent supply of FGFs.
FGF4, 8, 9, 17. These FGFs maintain the rapid proliferation and undifferentiated state of the underlying mesenchyme, allowing the limb to push outward.
And the classic evidence for this is surgical removal.
It is. If you surgically remove the AER, the limb stops growing immediately, and you get a truncation of distal structures. The earlier you remove it, the more proximal the remaining structure is. Conversely, an FGF-soaked bead implanted on the stump can completely substitute for the AER and rescue the outgrowth.
But outgrowth is only part of the story. The cells closest to the AER most recently formed the distal structures, like the hand or foot, while those that left the AER signal earliest formed the proximal structures, the shoulder or hip.
And the signal defining that proximal region comes from the adjacent trunk. Retinoic acid, or RA, diffuses from the main body axis into the limb base, and this induces proximal identity via the expression of genes like MYZ1 and MYZ2. RA is like the starting point marker, specifying the shoulder or hip.
Okay, moving to axis 2. Antero-posterior, AP patterning. This is the axis that controls digit identity, making sure you don't end up with 5 thumbs.
Right. This positional information resides in the zone of polarizing activity, or ZPA, which is a small, crucial signaling region located at the posterior margin of the limb bud.
And the signal coming from the ZPA is, once again, our friend Sonic Hedgehog.
It acts as a classic morphogen here, meaning its concentration determines the cellular response. It's released from the ZPA and diffuses to establish a gradient, with the highest concentration posterior and the lowest anterior.
And this concentration gradient dictates digit feet.
Exactly. High concentrations of specify posterior digits, like the pinky. Lower concentrations specify anterior digits, like the thumb.
And this was beautifully demonstrated by that textbook ZPA grafting experiment.
Right. If you take a ZPA from a donor embryo and graft it to the anterior margin of a host chick limb bud, you establish a secondary Sturr gradient. And this causes a spectacular mirror image duplication of the digits. You get like a 4-3-2-2-3-4 pattern.
Crucially, the AER with its FGF and the ZPA with its sh are not independent.
Not at all. They are locked in a positive feedback loop. They're like two partners maintaining a trade agreement. Shish from the ZPA maintains FGF expression in the AER, and FGF from the AER maintains shish expression in the ZPA. This mutual reliance is the engine that ensures sustained outgrowth and continuous patterning.
Also highly important in the AP axis is the repressive role of JLE3.
Yes, a component of the hedgehog pathway. JLE3 acts predominantly as a repressor, antagonizing target genes in the anterior region where SHE is weakest. The balance between SHESH and JLE3 is paramount. Loss of GLE3 function causes a common defect called polydactyly too many digits because the sh signal is unopposed.
And this pattern is further refined by the nested expression of the Hoxt genes.
HOXD9 to HOXD13, right across the AP axis. The general thinking is that the Schurz gradient sets the boundaries for their expression, giving each segment its final identity.
Finally, we address axis 3, dorsoventral DV patterning, which determines the difference between the dorsal surface, the knuckle side, and the ventral surface, the palm side.
And we know this pattern is defined by the epidermis, not the mesenchyme, because early inversion of the epidermal covering flips the internal skeletal and muscle pattern.
The default state appears to be ventral. Engrailed 1, or EN1, is expressed in the ventral epidermis.
And one of its key roles is to repress the dorsal signal, preventing Wnt7a expression on the ventral side while also limiting the physical extent of the AER.
So the dorsal determinant is Wnt7a, expressed exclusively in the dorsal epidermis.
And the experimental evidence for Wnt7a is very strong. If you lose the Wnt7a gene, the result is a double ventral paw. If you overexpress Wnt7a, you get a double dorsal limb.
So Wnt7a confers dorsal identity on the underlying mesenchyme by turning on the limb homeobox gene LMX1.
Exactly. And this creates a fundamental boundary. The dorsal LMX1 expressing mesenchyme and the ventral mesenchyme form a true compartment boundary. Cell lineage tracing shows that cells originating in the dorsal compartment never cross into the ventral compartment. They maintain a strict lineage segregation along this axis.
It is truly stunning that 3 separate integrated signaling centers, the AERFGF for PD, the ZPASH for AP, and the dorsal epidermis Wnt7a for DV, are all required and interlocked to build one coherent three-dimensional limb.
The integration is absolutely key. If any of the players fail to maintain the others, if Sh fails to maintain FGF or Wnt7a fails to maintain the AER, the whole system just collapses, leading to truncation or malformation.
With the structural elements of the trunk and limbs established, we now pivot to the final major components derived from the lateral plate mesoderm, the vascular and cardiac systems, essential for circulation and life support.
The development of the entire circulatory system is known as hematopoiesis, and it begins incredibly early. It also transitions through several distinct anatomical sites throughout development.
The first phase is primitive hematopoiesis, which starts in the yolk sac.
Right, and this phase produces a very specific type of blood cell, these large nucleated erythrocytes that contain embryonic hemoglobin. It's a temporary population.
And this is followed by definitive hematopoiesis, which creates all the adult blood and immune cell types, including the self-renewing hematopoietic stem cells.
And while both primitive and definitive lineages require 3 core transcription factors, GATA1, SEL, and LMO2, the definitive cells, the ones responsible for the adult system, absolutely depend on the factor RUNX1.
And the origin of these definitive blood cells was controversial for a long time, but it's now conclusively traced to the endothelium, the inner lining of the central embryonic arteries, particularly the dorsal aorta.
In a region known as the AGM, or aortic gonad mesonephros region, the key insight here is that blood progenitors don't originate as free-floating cells. They delaminate directly from the vessel lining, from a specialized population known as hemogenic endothelium.
And this was verified beautifully using molecular tracing techniques.
Yeah, the VE-cadherin-Cre system in mice, for example, which confirms that cells expressing endothelial markers later transition into and contribute to the hematopoietic stem cell population.
These definitive progenitors then embark on a crucial migration path.
They do. From the dorsal aorta, they travel to the fetal liver, which becomes the main hematopoietic center during the middle of gestation. And finally, they seed the bone marrow, which is the source of the adult hematopoietic system for the rest of our lives.
Okay, so parallel to blood formation is vessel formation. We define vasculogenesis as
the de novo, or new formation of vessels from progenitor cells in the lateral plate and extraembryonic mesoderm. This creates the initial primary capillary network.
And then later we have angiogenesis,
which is the process of generating new capillaries by sprouting, division, and movement from those existing vessels, expanding the network as the organs grow.
And a fascinating discovery is that arteries and veins are intrinsically different from the moment they are specified.
They are. They express complementary molecules that are essential for later capillary bed formation and fusion. Arterial endothelial cells produce the molecule ephrin B2, while venous cells express the receptor EphB4. These molecules act like a molecular handshake, guiding the proper connection between the two systems.
And the major factor driving vessel branching angiogenesis is VEGF, vascular endothelial growth factor.
VEGF stimulates endothelial cells to organize and form these highly polarized structures known as tip cells, which lead the growing sprout.
And this process of deciding which cell becomes the leader and which remains the stalk is a textbook example of lateral inhibition, governed by the Notch signaling system.
It is. The cell expressing the highest level of delta 4 becomes the tip cell. It then uses its Notch signaling ability to suppress its immediate neighboring cells from also becoming tip cells, ensuring sparse, controlled, and efficient branching.
This coordinated control prevents the entire vessel from just erupting in chaotic simultaneous sprouts. The inhibition ensures that only one highly specialized cell leads the way.
And in tying this system to another major developmental process, blood vessels and nerves often track together in the body. And this is not a coincidence.
Why does that happen?
It's because VEGF also binds to neuropilins, which are receptors for semaphorins, key molecules in neural guidance. This molecular link ensures that neural and vascular guidance cues are integrated. Nerves often secrete VEGF, attracting vessel growth to their location, ensuring that newly developed organs receive both innervation and blood supply simultaneously.
Okay, finally, let's turn to the heart, which originates from the splanchnic lateral plate mesoderm.
Yes, the early structure is the cardiac crescent, a horseshoe-shaped field of committed mesodermal cells. The induction of this crescent is critical and depends on signals from the underlying anterior endoderm, specifically BMP and FGF8.
And transcription factors like NK by 2.5 and TB by 5 are critically important in these early specification steps.
They are. Failure to form the heart is often caused by a lack of these endodermal signals. If the anterior endoderm is surgically removed, heart development is prevented entirely. Once induced, the two bilateral heart rudiments must migrate toward the midline and fuse into a single structure.
And if that fusion fails, the result is the rare but severe congenital anomaly cardia bifida, where two separate hearts form side by side in the chest cavity.
The fused structure is a simple tube that immediately starts beating. It must then undergo a massive morphogenetic event called looping, where the tube bends and folds to the right to establish the characteristic C or S shape of the future heart. This entire process is dependent on the embryo's overall left-right asymmetry system, particularly the expression of signaling molecules like nodal.
But that simple tube is not the entire heart.
No, it forms the atria and the left ventricle, which is known as the first heart field. The rest of the structure, and arguably the more complex parts, primarily the right ventricle and the outflow tract, which separates the aorta and pulmonary artery, is built later.
And this later growth comes from the recruitment of additional tissue from the second heart field, or SHF.
Right, a population of progenitors characterized by the expression of the transcription factor islet-1. These cells migrate anteriorly and posteriorly to integrate with the initial tube, ensuring that complex four-chambered structure can be completed.
And to achieve that final four-chamber structure, complex septation and remodeling must occur internally.
Key to this process are the endocardial cushions, which are masses of tissue that form at the atrioventricular junction. They are formed through an epithelial to mesenchymal transition of endocardial cells, induced by TGF-beta signaling from the surrounding myocardium. The cushions are essential. They eventually fuse to form the septum intermedium and remodel into the four heart valves.
Meanwhile, the muscular ventricular septum grows upward to separate the right and left ventricles, regulated by the TBX transcription factors like TBI5 and TBX20.
And concurrently, the tricky job of separating the two great arteries, the aorta and the pulmonary artery, occurs in the outflow tract. This region is septated by a distinct, specialized population of migrating neural crest cells, which travel from the head region down to the heart. This migration and integration is regulated by the transcription factor TBX1.
This entire highly choreographed process of remodeling is fraught with potential errors, which is why congenital heart defects are the most common birth anomaly in humans.
They are. Many human heart problems, like defects in looping, atrial and ventricular septa, or the infamous tetralogy of Fallot, which is often caused by defects in the neural crest-derived outflow tract septum, are associated with dominant mutations in those core transcription factor genes, like NKX2.5 and TBX5.
For instance, mutations in TBX5 cause Holt-Oram syndrome,
which strikingly involves specific defects in both the heart septa and the upper limbs, the same region where TBX5 is also required for forelimb patterning. Remember, TBI5 specifies forelimbs. This striking clinical connection vividly demonstrates how crucial these master regulatory genes are across multiple, seemingly unrelated mesodermal organ systems. Hashtag tag outro.
Hmm.
Here's where it gets really interesting. If we just step back from all the molecular complexity for a moment, the development of all these intricate mesodermal organs, bone, muscle, kidney, heart, it's simply a brilliant recurring exercise in precise cellular communication and decision-making.
It really is. We saw how gradients set spatial boundaries, and FGF patterning the limb, high to low. We saw how negative feedback loops create a temporal rhythm, the Hes clock defining somite segments with minute-by-minute accuracy.
And we saw how reciprocal inductions enforce these necessary partnerships,
Yeah.
the ureteric bud and the mesenchyme cooperating to build the kidney filter. The Wents, the BMPs, the FGFs, the Hox genes, they're the common ancient language used throughout the embryo to build structure.
And as you pointed out, errors in these fundamental molecular circuits are directly responsible for so many congenital anomalies in humans, often because the organism can't function with half the normal gene dosage, a fragile state known as haploinsufficiency.
It's true.
And that brings us to a final provocative thought for you to mull over as you wrap up this deep dive.
Well, while we as adult mammals can repair our skeletal muscles using those PAX7 positive satellite cells, we lose the ability to regenerate complex organized organs, like the heart and kidney shortly after birth. Instead, we just scar.
But lower vertebrates like salamanders and zebrafish, they retain this full regenerative capacity for life. They can regenerate a fully functional heart or kidney structure after an injury.
Precisely. This means the underlying developmental machinery, the molecular ability to signal, recruit progenitors, and rebuild the whole structure, must persist at some level in our cells. So the profound question in regenerative biology isn't just how do we turn the developmental process back on, but why did higher organisms evolve to silence this powerful regenerative capacity in favor of permanent scarring after that critical postnatal window?
Understanding that evolutionary trade-off, why we chose structural stability over regenerative potential, that seems to be the key to unlocking future therapies for organ failure.
It absolutely is. It's a fascinating question. Thank you for taking this deep dive into the blueprint of mesodermal organ development with us. We hope you feel thoroughly informed and perhaps a little inspired by the molecular elegance of how complex life emerges from these few simple, repeatable rules.